Ever get the feeling your body's got this whole, like, secret language going on beneath the surface?
Uh-huh.
Like a hidden network constantly sending messages you can't quite hear.
Yeah, that's a great way to put it, actually.
Well, it turns out that feeling isn't too far off. Today, we're diving deep into the world of exosomes.
The body's tiny messengers.
Exactly. Think of them as the body's miniature postal service. Tiny packages cells use to talk to each other.
Hmm.
And for those of you listening who want to get smart on important topics, but like without getting totally bogged down in jargon, you are definitely in the right place.
We aim to please.
We're going to unpack what these little messengers are all about, what they actually do, and why their communication skills are so, well, unique.
And it's important stuff. This isn't just, theoretical science. It has real implications for understanding health and even developing new therapies down the line.
Absolutely. So in this deep dive, we're tackling some key questions, right?
We are, first off, what are exosomes? In simple terms, break it down. Second, what kinds of jobs do they actually handle within the body? What's their role?
Right.
And third, what makes them stand out? What's unique about their role in the body's whole communication system compared to other ways cells chat?
Got it. Because these tiny vesicles, they sound more significant than maybe their size suggests.
Oh, definitely. They offer really valuable insights into our well-being and, like I said, the development of new medical treatments.
Okay, so let's get right to it then. What exactly are these exosomes? When I hear tiny messengers, I still picture like microscopic FedEx guys running around.
That's a fun image. Think of them more like nano-sized bubbles, really, really small.
Nano size.
Yeah, far too small to see, even with a regular microscope. We're talking like 30 to 150 nanometers across.
Wow, okay. Put that in perspective for us.
Well, okay, lipoproteins, the things that carry fats in your blood. Exosomes are about that size.
Right.
But they're incredibly smaller than any of your actual cells. Like, imagine a cell as a basketball. These exosomes, they'd be like tiny specks of dust floating around it.
Dust-sized messengers. Okay, so where do these little specks of information even come from? Is there like a cellular factory churning them out?
Sort of, yeah. They're formed inside cells through this really fascinating process. Picture a cell having internal sorting offices.
Okay, sorting offices.
These are called late endosomes. And sometimes they mature into these larger compartments known as multivesicular bodies, or MVBs.
MVBs, got it.
Now, within these MVBs, even smaller bubbles start to form. These are the intraluminal vesicles, ILVs. Think of them as baby exosomes getting packaged up. The key moment happens when these MVBs travel to the edge of the cell, the plasma membrane, and they fuse with it.
And release the babies.
Exactly. They release those inner bubbles, now fully fledged exosomes, out into the environment around the cell.
Huh.
And interestingly, they're not just floating free. They can also get stored within the tissue matrix itself. And we even see cells in lab dishes releasing them constantly.
So these little bubbles, the exosomes, what kind of information are they actually carrying? What's the message inside the envelope, so to speak?
This is where it gets really interesting. They're packed, and I mean packed, with a diverse collection of biological molecules, stuff they've basically inherited from the cell they came from.
Like what kind of stuff?
Well, think of it like those dust-sized bubbles are stuffed with all sorts of cargo. You've got proteins in there, proteins that do everything from building cell structures, acting as signals, enzymes, growth factors.
Oh, yeah, little proteins.
Yeah. And various fats or lipids. Plus, and this is really key genetic instructions too, things like DNA fragments and different types of RNA.
Wait, RNA too? Like mRNA?
Yep, mRNA, miRNA, which is microRNA. So this diverse cargo is what allows them to have such a wide range of effects on the cells that receive them.
So it's not just proteins, but lipids, DNA, RNA. That's quite the care package. Anything else tucked inside these cellular delivery vans?
Well, the lipids themselves are important things like cholesterol, specific lipid structures called RAFs, ceramides.
Yeah.
And as I mentioned, those nucleic acids, DNA, mRNA, miRNA, even long non-coding RNA. So they're not just, ferrying around building blocks. They have the potential to deliver actual genetic blueprints or regulatory molecules.
This is wild. So one cell can basically send out a bubble to another cell containing proteins, fats, and instructions for making more proteins. It's like a cellular software update service.
That's a great analogy, yeah. Kind of is.
Now, I think you mentioned earlier that scientists are still figuring out if exosomes are truly unique compared to other Extracellular vesicles. What's the deal there?
Yeah, that's a really crucial point to make clear. While we're focusing on exosomes today, they are part of a larger family.
Okay.
The family is called extracellular vesicles, or EVs. And there are different types of EVs formed in different ways.
Right.
So the scientific community is still actively researching, do exosomes have truly distinct characteristics and functions that set them apart from all the others? Or are the main differences really about their specific size range and that internal formation pathway within the MVBs?
So the jury's still out a bit on their absolute uniqueness within the EV family.
Pretty much. It's an area where our understanding is constantly evolving. As the research progresses, definitions get refined.
Okay, that makes sense. So we've got these tiny cargo-filled bubbles being released by cells. What happens next? How do they actually communicate with other cells?
Right. So their main job is that intercellular communication. Once they're released, these exosomes can travel sometimes just next door, sometimes quite a distance away through fluids. And they influence those recipient cells in a couple of main ways. They can either physically merge with the recipient cell or be taken up by it, delivering their contents directly inside.
So dropping off the package.
Exactly.
Yeah.
Or they can simply bump into the recipient cell's surface, interact with receptors there, and activate signaling pathways without actually going inside.
Kind of like ringing the doorbell to get a response.
Precisely. Both methods work to pass the message along.
And this communication, is it happening like all the time in our normal, healthy bodies? Or is it more involved when something specific is going on, like an injury?
It's definitely a constant process. It's happening in normal, everyday biological functions, absolutely.
Yeah.
But it also ramps up or changes during specific events, like when the body needs to heal or fight off an infection or deal with inflammation.
Okay.
We're discovering that exosome communication plays a role in a surprisingly broad range of activities. Things like angiogenesis, that's the growth of new blood vessels.
Right. Important for healing.
Very. Also, the process of wound repair itself, managing inflammation, turning it up or down, and even helping cells migrate, move around the body.
So how does a cell actually release these things? You said the MVB fuses with the membrane, but is there a specific mechanism? It sounds pretty coordinated.
Oh, it's very well orchestrated. It involves several key players inside the cell. You've got the cell's internal skeleton, the cytoskeleton,
Okay.
made of proteins like actin and microtubules. These act like tracks.
Cracks for the packages.
Exactly. Then you have tiny molecular motors, proteins like myosins and kinesins, that physically move the MVDs, those packages containing the baby exosomes, along these tracks towards the cell's outer edge.
Right.
And there are specific guiding proteins, small GTPases called RAB proteins, that help make sure these MVBs get to the right docking spot on the plasma membrane.
Like a cellular GPS system.
Sort of, yeah. And then finally, there's specialized machinery involving proteins called snares that helps the MVB membrane actually fuse with the cell's outer membrane, popping the exosomes out into the extracellular space.
It really is like a carefully choreographed cellular dance.
It is quite intricate.
You also mentioned something called the ESCRT machinery earlier. That sounds like another key part of this whole process.
Absolutely. ESCRT, that stands for endosomal sorting complex, required for transport.
Catchy name.
Yeah. This machinery is a major player in the initial formation of those inner vesicles, the ILVs, inside the MVBs.
So making the baby exosomes in the first place.
Precisely. Think of ESCRT like a miniature factory line within the cell, specifically designed for sorting cargo into what will become exosomes and helping the membrane bud inwards.
How does it work?
Well, it's actually a complex of different protein groups, ESCRT0, IE33, and another helper called VPS4. They work together in sequence. Some parts recognize and gather the specific molecules, the proteins, the RNA destined for the exosome.
Okay, grabbing the cargo.
Right. Other parts then help the membrane of the MVB start to curve inwards, pulling that cargo with it, and eventually they help pinch off that inward bud to form the vesicle, the ILV. It's like molecular origami, really.
Wow. So there's this whole dedicated system just for packaging and eventually releasing these tiny messengers. But you also hinted that there might be other ways exosomes can form, maybe without this ESCRT machinery.
That's right. While the ESCRT-dependent pathway is definitely a major route and maybe the most studied, research has uncovered that cells can also make these intraluminal vesicles and thus exosomes through ESCRT-independent mechanisms.
Really? Like how?
Well, one way involves lipids, specifically the production of a lipid called ceramide by an enzyme, sphingomyelinase. It seems that generating ceramide in the membrane can actually help it curve inwards and form vesicles.
Interesting. So fats can help bend the membrane.
It appears so. Also, certain proteins that are really abundant on the surface of exosomes called tetraspanins. You might see names like CD63, CD9, CD81.
Yeah.
These tetraspanins, along with other lipids like cholesterol and phosphatidic acid, have also been implicated in organizing membrane domains and potentially facilitating vesicle formation independently of ESCRT.
So the cell has a few different tricks up its sleeve for making these vesicles.
Seems that way, yeah. It gives the cell flexibility, perhaps for packaging different types of cargo or releasing exosomes under different conditions.
This is truly fascinating. We're getting a great picture of what exosomes are and how they're made and sent out. Now let's dig into some of the really unique roles they play. You mentioned they do more than just simple signaling.
Precisely. Their unique cargo and delivery mechanism allow them to participate in some really fundamental and sometimes surprising biological processes.
Like the genetic material transfer you mentioned.
Exactly.
Yes.
Because they carry things like messenger RNA, mRNA, and microRNA, mRNA, they have this remarkable ability to actually alter gene expression, which proteins get made in the cells that receive them.
How does that work, say, with mRNA?
Well, a microRNA delivered by an exosome can enter the recipient cell, find a matching mRNA molecule, which is the blueprint for a protein, and bind to it. Often, this binding leads to that mRNA being degraded or blocked from being translated into a protein.
So one cell can remotely dial down the production of a specific protein in another cell.
That's exactly it. It's like cells are sharing regulatory software updates, not just data.
That's incredible. And you also mentioned something earlier about a possible influence on traits being passed down. Epigenetics.
Yes, this is a really cutting edge and frankly quite speculative area, but it's intriguing. The idea builds on that RNA transfer. Epigenetics involves changes in gene activity that don't change the DNA code itself, right?
Right, like environmental influences turning genes on or off.
Exactly. And sometimes these epigenetic changes can potentially be inherited across generations. The fact that exosomes can deliver molecules like miRNAs, which do regulate gene activity, raises the fascinating, though still debated possibility that they could be one mechanism contributing to this extra layer of biological inheritance, maybe transmitting acquired traits or responses.
Oh, okay. That's mind-blowing stuff. Needs more research, I imagine.
Oh, absolutely. A lot more research, but it opens up some profound questions.
Definitely. So what other unique roles? You mentioned reproduction earlier.
Yes, they play some critical roles there. In male reproduction, for instance, as sperm mature in the epididymis, they receive exosomes from the surrounding cells. These exosomes transfer functional proteins and maybe RNAs that are essential for the sperm to become fully mature, motile, and capable of fertilization.
So they're helping equip the sperm for their mission?
You could say that, yeah. And in pregnancy, it's fascinating. The placenta releases large amounts of exosomes into the mother's circulation.
Okay.
These placental exosomes are thought to be crucial for establishing and maintaining maternal immune tolerance towards the fetus. Remember, the fetus has paternal antigens, making it technically foreign to the mother's immune system.
Right, so the exosomes help prevent rejection.
They seem to play a big part in modulating the mother's immune response at that crucial interface, preventing an attack on the fetus. Although it's complex depending on their specific origin and cargo, placental exosomes might have different effects.
Still, a vital role in something so fundamental. What about when things go wrong, like tissue damage? Do exosomes help with repair?
Absolutely. This is a huge area of therapeutic interest. Exosomes derived from stem cells, particularly mesenchymal stem cells, or MSCs, have shown tremendous promise in promoting tissue repair and regeneration.
How do they do that?
Well, stem cells are known for their healing capabilities, and it turns out that a significant part of their effect isn't just them turning into new cells, but it's the signals they release, often packaged into exosomes.
Ah, so it's the messages they send out.
Exactly. These MSC-derived exosomes can deliver regenerative signals, growth factors, anti-inflammatory molecules, RNAs directly to damaged tissues. They can encourage local cells to proliferate, reduce inflammation, promote the formation of new blood vessels, basically orchestrate the healing process.
So like sending out tiny repair kits and instructions.
That's a great way to think about it. There's a lot of preclinical work, for example, using MSC exosomes for things like bone fracture healing, where they've been shown to improve bone regeneration and blood supply to the healing area.
Any specific examples?
Yeah, some studies have shown that pre-treating the stem cells, maybe by exposing them to low oxygen, can actually boost the regenerative potential of their exosomes, possibly by increasing the amount of certain beneficial miRNAs, like miR-126, which promotes blood vessel growth.
Wow. So you can potentially tune the exosomes.
That's the idea researchers are exploring, yes. Using different sources of stem cells, like from umbilical cords, or pre-treating them in specific ways, or even directly loading exosomes with specific molecules like miR-29A to enhance their effects on things like bone healing or angiogenesis.
Fascinating. Okay, what about our defense system, the immune system? Are exosomes involved there too?
Deeply involved. Immune regulation is another major function. Exosomes act as crucial messengers between different immune cells, helping to coordinate the entire immune
Oh,
response.
so
They can carry antigens, for example, to help activate specific immune cells to fight an infection, but they can also carry signals that suppress immune responses.
why would they do that?
Well, it's essential for maintaining balance, preventing the immune system from overreacting and causing autoimmune diseases or chronic inflammation. Exosomes from certain immune cells, like regulatory T cells, can carry immunosuppressive signals.
So it's a communication network for both activating and calming down the immune system.
Exactly. It's a very dynamic interplay. Of course, this can be exploited in disease too. For example, tumor-derived exosomes can carry signals that actively suppress the immune cells trying to attack the tumor.
Ah, a dark side to the immune role.
Unfortunately, yes. And in autoimmune diseases like multiple sclerosis, exosomes are thought to be involved in the processes of inflammation and damage to the nervous system. But conversely, MSC-derived exosomes are being studied as a potential therapy for MS, precisely because they can have those anti-inflammatory and immunomodulatory effects.
It seems like exosomes are just everywhere. What about our brains? Do these tiny messengers operate in the central nervous system?
They absolutely do. Communication between neurons and glial cells, the supporting cells in the brain, involves exosomes. This is critical for normal brain function, synaptic plasticity, things like that. But similar to the immune system, there's also a link to disease. In neurodegenerative conditions like Alzheimer's and Parkinson's, there's growing evidence that exosomes might, unfortunately, play a role in spreading the misfolded pathological proteins associated with these diseases, like tau and amyloid beta in Alzheimer's, or alpha-synuclein in Parkinson's from cell to cell.
So they could be helping the disease progress.
That's a major hypothesis being investigated, yes. It's like they're unintentionally delivering toxic cargo.
Yikes. Is there a therapeutic angle here too?
There is definitely. Researchers are exploring using beneficial exosomes, perhaps from neural stem cells, NSCs, to treat neurological damage. For example, studies in stroke models have shown that NSC-derived exosomes might promote the birth of new neurons, reduce inflammation, and improve outcomes by delivering supportive factors. Some studies have even tried loading NSC exosomes with specific growth factors, like BDNF, and found better results in stroke models. There's also work looking at stem cells from dental pulp and their exosomes, which showed some promise in stroke models too.
It's almost hard to keep track of all the roles. You also mentioned wound healing specifically earlier.
Yes, cutaneous wound healing skin wounds. Again, MSC-derived exosomes are a hot topic here. They seem to positively influence all the major stages of wound healing, reducing the initial inflammation, promoting the proliferation of skin cells and angiogenesis for new blood supply, and helping with the final remodeling of the tissue.
Are there specific molecules involved?
Yes, researchers are identifying specific cargo. For instance, exosomes from adipose-derived stem cells containing a long non-coding RNA called MALAT1 were shown to help healing by regulating a specific microRNA. Another study found that exosomal miR-135A from amniotic membrane stem cells could promote wound healing and get fibroblasts moving. It's about delivering the right signals at the right time.
Incredible specificity. Now, we touched on the darker side with cancer and immune suppression. Can you elaborate on their role in cancer progression?
Yeah, unfortunately, cancer cells seem to hijack this communication system very effectively for their own benefit. They release large quantities of exosomes that profoundly shape the tumor microenvironment
How do they shape it?
in multiple ways. These tumor-derived exosomes can carry factors that stimulate angiogenesis, bringing more blood supply to the tumor. They can carry enzymes that break down the surrounding tissue, making it easier for cancer cells to invade. They can transfer molecules that promote EMT, that's epithelial mesenchymal transition, a process that makes cancer cells more mobile and invasive.
So helping the tumor grow and spread.
Yes, and also helping it survive. They can transfer drug resistance mechanisms between cells, making treatments less effective. And crucially, they can travel to distant sites in the body and help prepare those sites create what's called a pre-metastatic niche, making it easier for stray cancer cells to land there and form secondary tumors or metastases.
Wow, so they're actively paving the way for metastasis.
It seems that way in many cases, yes. They're really key players in tumor development, spread, and resistance.
It really highlights the double-edged sword nature of these vesicles. But let's swing back to the positive potential you mentioned, drug delivery.
Right. This is one of the most exciting therapeutic applications being explored. Because exosomes are natural, produced by our own bodies, they have some inherent advantages as potential drug delivery systems.
Such as?
Well, they tend to be biocompatible and have low immunogenicity, meaning they're less likely to provoke an unwanted immune reaction compared to, say, synthetic nanoparticles.
Okay, that's a big plus.
And crucially, they seem to have an innate ability to cross biological barriers that are notoriously difficult for many drugs to penetrate. The big one being the blood-brain barrier.
Getting drugs into the brain.
Exactly. That's a huge challenge in treating neurological diseases and brain cancers. Exosomes offer a potential natural pathway to get therapeutics across that barrier.
So how do you get the drug into the exosome?
That's the trick researchers are working on. There are various methods being developed for loading therapeutic cargo, small molecule drugs, proteins, therapeutic RNAs like siRNA or miRNA into exosomes. This can be done passively, just by incubating the exosomes with the drug, or actively using techniques like electroporation or genetic engineering of the parent cells to package the desired cargo.
And can you target them? Make them go to specific places?
That's another key area of research-targeted delivery. Scientists are trying to engineer the surface of exosomes, perhaps by adding specific proteins or antibodies, so that they preferentially bind to and deliver their cargo only to the target cells, like cancer cells, while sparing healthy tissues.
So smart, targeted delivery using the body's own system.
That's the goal. There are already preclinical studies and even some early clinical trials exploring this potential in various diseases, including brain cancer and conditions like acute respiratory distress syndrome.
ARDS.
Very promising. And finally, you also mentioned they could be used as biomarkers, like indicators of disease. Yes, precisely. Because exosomes are shed by cells, including disease cells, and circulate in bodily fluids like blood, urine, saliva, they carry a molecular fingerprint, a proteotranscriptomic signature of their cell of origin.
Meaning they contain proteins and RNA specific to that original cell.
Exactly. So by isolating exosomes from a readily accessible fluid like blood and analyzing their contents, the specific proteins, RNAs, even DNA fragments they carry, we might be able to detect the presence of diseases like cancer much earlier than current methods allow.
Like a liquid biopsy.
Essentially, yes, a non-invasive way to get information about what's happening in tissues deep inside the body. They could also potentially be used to monitor disease progression or a patient's response to treatment. There are clinical trials underway right now evaluating exosomes as diagnostic and prognostic markers for various cancers and other conditions.
This is all incredibly detailed and really highlights their versatility. So to really drive home their significance, can we specifically talk about what exosomes can do in addition to, or maybe better than, other ways cells communicate? We know cells have other methods, right? Like direct contact or just releasing simple molecules.
That's a really great question to focus on because, yes, cells talk in many ways, but exosomes offer some unique advantages.
Okay, like what?
Well, first, think about the complexity of the cargo. Unlike simple signaling molecules binding to a receptor, or even direct cell junctions transferring limited stuff, exosomes can package and deliver a whole cocktail of diverse molecules, simultaneously proteins, lipids, multiple types of RNA directly into the recipient cell's cytoplasm or nucleus.
So it's not just one signal, but a whole package of instructions and tools arriving at once.
Exactly. It's a much more complex and potentially nuanced message than just, say, releasing a single growth factor.
Okay, what else?
Second, the protection aspect is huge. The exosomal membrane, that lipid bilayer bubble, shields its cargo from degradation in the harsh extracellular environment.
Protecting the message.
Critically important, especially for fragile molecules like RNA, which would be rapidly chewed up by enzymes called nucleases if they were just released freely. Exosomes act like little armored delivery vans.
Got it. Secure delivery. What about distance?
That's another key differentiator. While some signals diffuse locally, exosomes can travel through bodily fluids, blood, lymph, cerebrospinal fluid, to reach cells and tissues far away from their origin. This enables true long-distance systemic communication throughout the body.
So they're built for cross-country communication, not just neighborhood gossip.
Exactly. They can coordinate responses between distant organs in a way that simple diffusion can't.
Okay, complex, cargo, protection, long distance. Anything else?
Yes, the potential for modulating multiple pathways simultaneously. Because of that diverse cargo, a single exosome landing on or entering a cell can trigger changes in several different signaling pathways or cellular processes at the same time.
Leading to a more complex response.
Potentially, yes. A more integrated or multifaceted response in the recipient cell compared to activating just one pathway with a single ligand.
Makes sense. And what about getting into tricky areas? You mentioned the blood-brain barrier.
Right, that's a big one. Their small size, combined with specific proteins on their surface, seems to allow some exosomes to cross biological barriers that are impermeable to most molecules and even larger particles. The blood-brain barrier is the classic example.
So unique access.
Unique access, which is vital for communication within protected compartments like the brain, but also offers that huge therapeutic potential for delivering drugs to those areas.
Okay, that's a clear advantage. And finally, let's revisit that epigenetic idea, the RNA transfer. That seems pretty unique compared to standard signaling.
Absolutely. The ability to transfer functional RNAs like mRNA and miRNA directly into another cell provides a mechanism to influence the recipient cell's gene expression program, potentially in a stable long-term way, without altering the DNA sequence itself.
Changing cell behavior from the inside out, essentially.
Yes, and that raises those profound, though still speculative possibilities about influencing cell fate, function, and maybe even contributing to non-genetic inheritance patterns. It's a mode of communication with potentially deeper and longer-lasting consequences than just transient signaling.
Wow. Okay, putting it all together like that really emphasizes why there's so much excitement around exosomes. They're not just another messenger. They have these distinct capabilities.
They really do. They represent a very sophisticated layer of biological communication.
This has been an absolutely fascinating deep dive. It's incredible to realize how these minuscule vesicles are tangled up in so many fundamental processes, from reproduction to immunity to brain function, and hold such promise, but also some challenges for medicine. Where is the research heading now? What are the next big steps?
Well, the field is moving incredibly fast. A lot of effort is going into optimizing that therapeutic potential we talked about.
The drug delivery part.
Yeah, refining methods for both actively and passively loading specific therapeutic materials, drugs, proteins, genetic payloads into exosomes efficiently, and also improving targeting, making sure they go exactly where we want them to go.
Makes sense. What else?
There's also growing interest in exosomes, or rather exosome-like nanovesicles derived from non-mammalian sources like plants.
Plant exosomes, really?
Well, exosome-like vesicles. They seem to exist, and researchers are exploring if they might have unique properties or advantages, maybe for oral delivery or in terms of cargo. It's an emerging area.
Interesting. And clinically?
Clinically, we're likely to see continued development and hopefully more validation of exosomes as those biomarkers we discussed for earlier diagnosis, monitoring disease, predicting treatment response. And of course, the ultimate goal is translating exosome-based therapies into treatments for patients across a wide range of diseases, orthopedic, neurological, cancer, inflammatory conditions.
But there are hurdles, right?
Oh, definitely. Big challenges remain. Things like reliably manufacturing large quantities of clinical-grade exosomes. Upscaling production is tough. Establishing standardized methods for isolating and characterizing them, so results are comparable between labs, and navigating the regulatory pathways for these novel therapeutics. These are all significant hurdles the field is actively working on.
So huge potential, but still important groundwork being laid. Still, it definitely gives you that real aha moment, doesn't it? Understanding that exosomes represent this incredibly sophisticated, versatile communication network humming away inside us, with implications reaching far beyond what we might have initially guessed.
Exactly. They're not just a minor detail. They seem pretty fundamental to how multicellular organisms function and coordinate themselves.
Well, that brings us towards the end of our deep dive into the world of exosomes.
So if we were to sum it all up, what's the big takeaway for everyone listening?
I think the big picture is that these minuscule, nano-sized vesicles are anything but insignificant. They are powerful, multifaceted communicators. They're constantly shuttling this incredible wealth of information, proteins, lipids, RNA between cells, performing unique feats of messaging that go way beyond just simple signaling. They protect their cargo, travel long distances, cross barriers, and can even reprogram cells from the inside.
It really highlights the complexity.
It truly underscores the incredible complexity and, honestly, the elegance of how our bodies function right down at the cellular level. And what's so exciting, I think, is that we're probably only just scratching the surface of what they can do.
Absolutely. It's a field just packed with possibilities for future discoveries.
It really makes you wonder, doesn't it? If these tiny exosomes are constantly exchanging information and influencing cells in all these interconnected ways, well, how might our understanding of seemingly separate processes in the body, like, say, metabolism and immunity, or gut health and brain function, become even more integrated in the future as we learn more about these messengers?
That's a great thought, connecting the dots.
Yeah, it's definitely something fascinating to consider for your own health and well-being. So if this deep dive has sparked your curiosity, and I hope it has, we really encourage you to maybe look for new research on exosomes, especially in areas that caught your interest today. Like you said, new discoveries are popping up all the time in this field.
Keep your eyes peeled.
Definitely. Until our next deep dive, keep exploring the amazing complexities working away inside you.